The endoplasmic reticulum. History. In 1945, Keith Porter and his associates observed a fine reticulum in cells of chicken fibroblast using electron microscopy.
Structure. The eor is a large membrane-bound organelle spread throughout the cytoplasm of eukaryotic cells. This organelle forms an interconnected network of flattened, membrane-enclosed sacs known as cisternae. The membranes of the eor are continuous with the outer nuclear membrane.
The eor provides 50% of the total membrane surface of the animal cell. It's an incredibly important structure in living cells. Let's take a closer look.
Functions. Rough endoplasmic reticulum. Protein production, protein folding. Smooth endoplasmic reticulum. Lipid production, detoxification, and storage of ions.
The rough eor. The outer nuclear membrane is studded with ribosomes and is continuous with the membrane of the rough endoplasmic reticulum. The average liver cell holds roughly 13 million ribosomes.
The rough eor derived its name from the ribosomes embedded on its outer surface. These ribosomes are membrane-bound and assemble proteins, which is also known as translation. Proteins are produced at this site for the plasma membrane, Golgi apparatus, secretory vesicles, lysosomes, endosomes, and the endoplasmic reticulum itself. It is in the rough eor that hemoglobin is produced, as well as glycoproteins.
Another very important aspect of the rough eor is that it's where proteins are folded into the lock and key shape that is responsible for recognition. The lumen of the rough eor is like the bouncer of the eor. It checks the quality of each protein, rejecting those that are folded or formed incorrectly. These rejected proteins are stored in the lumen or recycled and broken down into amino acids.
On the other hand, the smooth eor is responsible for very different functions. While the eor manufactures proteins, the S-eor manufactures lipids. The S-eor is an important structure for detoxification. Consequently, there are large amounts of S-eor in liver cells to break down toxins.
The S-eor has the ability to double its surface area within a few days, returning to its normal size when detoxification is no longer urgent.
Another function of the S-eor involves calcium. The sarcoplasmic reticulum is a variation of the S-eor. It stores ions and releases them when the body needs them. For example, calcium ions are released from the S-eor when our muscle cells need to contract.
As you can see, the eor is a very important organelle with many important functions. Cells specialising in the production of proteins will tend to have a larger amount of the rough eor, while lipid-producing cells and steroid hormones will have a greater amount of smooth eor.
The eor has domains that contact the plasma membrane, the Golgi, mitochondria, vesicles, and perioxysomes, the majority being part of the endomembrane system.
Evolution of the eor. It is believed that the plasma membrane invaginated the process of folding back on itself to form a pouch, to form many internal membranes. This then led to the eor. An intermediate form of this invagination can be seen in an organism with prokaryotic and eukaryotic characteristics. It suggested further elaboration of the eor may have given rise to the Golgi apparatus and the peroxysomal reticulum.
In conclusion, the eor is a very important organelle that carries out many functions which sustain life.
Created using Powtoon.